this fragment is amplified several times, this process is called bridge amplification, and there are clusters of amplified DNA on the flow cell. You can see. So for a good sequencing, these clusters should be separated from each other so that the machine can separately identify each cluster. So again, same DNA polymerase adds new bases which are complementary to the template strand. So these bases are fluorescently labeled. So as the new base is added, the machine detects which base has been incorporated. There are 4 types of bases. Each base has different fluorophores. One is green, one is blue, one is red, and one may be yellow. So depending upon the color, the machine detects which base has been incorporated at each cluster, and the machine records in this format. Each color is corresponding to one nucleotide. So this is just the image of what screen shows of my machine. So this is the quality score of newly synthesized DNA fragments. These are number of nucleotides, and this shows how good DNA quality is sequenced within the machine. So you can see here the quality score, which is also called the front quality score, is most of the sequencing reactions. The quality score is greater than 30. So I think I do not need to explain this format of FASTQ, which is just a text file which contains the DNA sequences. So each line describes one DNA sequence. The first line is the unique identity of the read. The second line is the DNA sequences. The third line is the plus sign, and the fourth line is the quality score of these sequences. So these 4 lines are repeated again and again and again in a single file. And this format is termed as FASTQ format of storing the DNA sequence. So now comes to the MinOIN. So this is the 3rd generation sequencing. So why this is the 3rd generation? Because the length of DNA fragments which can be sequenced is much higher than can be performed in 2nd generation sequencing like Illumina or INTORANT. So in Illumina, the maximum read length is approximately 600 base pair. But now in 2023, they have claimed an instrument which can produce long PCR, just like Oxford Nanopore. So they have collaborated with PacBio and just made a new machine, and they have introduced long range sequencing. But that is not a mainstream process right now. So in Oxford Nanopore, this is a MinOIN part of machine. So what happens? I think you guys are very well aware. So there is a nanopore in the membrane. DNA fragments pass through that nanopore, and as the DNA fragment passes through the nanopore, that is detected within the machine, the DNA sequence is recorded. So for this purpose, there is no need of DNA polymerase or no need of fluorescently labeled bases to be incorporated. The DNA is directly detected what is the sequence of the molecule. So for this purpose, the library preparation of Oxford Nanopore is a bit different from the Illumina, but more or less the steps are similar. So first step is the isolation of genomic DNA, and then we have to encrypt the DNA. So what happens during this encrypt? Usually the ends of double-stranded DNA which we produce through fragmentations are not even. Those are not blunt ends. Those are usually sticky ends. So using end prep, these sticky ends are produced to blunt ends, and then a nucleotide is added for adaptive ligation. So this is end prep of DNA, and then adapters are ligated. These adapters have a T overhang which make a complementary to this A, and the ligation efficiency increases to manifold. So this step is almost similar, which I have demonstrated previously for Illumina. Then we have to attach ligation. We have to ligate the sequencing adapters. What are these adapters? In case of MinOIN, these adapters carry specific proteins. Those proteins direct the DNA fragments to the nanopore on the membrane. So if we do not attach these fragments to our unknown DNA fragments, if we do not attach these adapters, our fragments will not pass through the nanopore which is present on the immunoid membrane. So for that purpose, we have to attach these adapters. These adapters contain some specific proteins. These proteins direct our fragments to the pores, and then DNA passes through the pore for real-time sequencing. So this is just a cartonic representation. This is our double-stranded DNA. So one strand is passed through this nanopore, and as this DNA passes, there is a variation in the voltage across the membrane. So this variation in voltage is different for four bases for the resolution of the sequences. So this is different for adenine, guanine, cytosine, and thymine. So each variation is recorded through a computer, and the sequence is recorded. So I think this is a very brief overview about the process how it will take place during the NDS Lumina and Oxford Nanopore.